We journey now into the fundamental unit of life. Your health depends on what is happening in the types of cells from which you are fashioned. In this lecture, I will cover the cell theory, both passive and active transport, structure of the cell membrane, architecture of the cell itself, and finally, the functions of cell organelles. You perhaps already know that all living things are made of one or many cells. However, the size of a plant or an animal depends not on the size of its cells, but on the number of cells which compose its body. The larger the animal or plant, the more cells it contains. In the lowest form of life, the whole organism is only a single cell, one in which are carried on all the processes associated with life. Such is a unicellular organism. In the higher forms of life, multiplied millions of cells arrange themselves into groups of different type cells in order to meet the varied physiologic needs of that organism. Such an organism with a division of labor is a multicellular one. Anthony van Leeuwenhoek of Holland first described microbes, bacteria and protozoa. He made his own microscopes, which were superior to any of that time. He is known as father of bacteriology because it was he who first accurately described the different shapes of bacteria. Coxal shape, as you remember from lecture number 5, coxy means berries in Greek. Bacillary shape means rods-like in Latin. And spiral shape. Spirochete, spiro stands for spiral. And chete is Greek, stands for long hair. Leeuwenhoek also pictured bacterial cell arrangement in infected material. Examples are streptococcus, remember it as the strips of cocci. Diplococcus, diplo in Greek means double. Staphylococcus, staphylo in Greek means bunch of grapes. Think of it as a clusters of cocci. Streptobacilli, strips means chains, so chains of bacilli. Or you can think of them as a strips of bacilli. It is easier. Streptobacilli, strips of bacilli. Leeuwenhoek was a wealthy man who devoted the major portion of his time to scientific studies. He fashioned 250 microscopes, grinding most of the lenses himself. Leeuwenhoek was obsessed with his microscopic findings and examined with his scopes everything he could think of. He is also known as father of scientific microscopy. He gave the first complete account of red blood cells. He discovered sperm cells. He demonstrated the capillary connections between arteries and veins, and he made other important anatomic observations. Available at that time, Kircher's microscope magnified only 32 times. The beta scopes fashioned by Leeuwenhoek magnified about 270 times. Another scientist and enthusiast, Robert Hooke, observed cork under a microscope and called the chambers cells. So let me tell you how this actually happened. Robert Hooke had been appointed curator of experiments for the Royal Society of London, and he knew he had to come up with something good for the next weekly meeting. He was only too aware that the elite of British science would be there, and he wanted to present a demonstration that would enlighten, entertain, and impress them. It wouldn't be an easy task. Hooke had an idea. Perhaps he would use an exciting new technology of 17th century, lenses, cast and ground of glass. The world was bustling with talk of lenses. With the pair of lenses in a frame, nearly blind were able to see. A miracle came true. Old men who had not been able to read for years had their books and letters restored to them. Early in the century, Galileo had pointed a lens to the sky and had started an intellectual revolution. The human eye has a voracious appetite, and Hooke knew it. But Hooke was himself interested in a new use of lens, to look at very small things. In fact, he had built his own microscope, one of the first in the world. So for scientific demonstration, Hooke thought of using the microscope to try to see why corks float. Cork was a mystery. It appeared to be solid, yet it floats. Perhaps it is not so solid after all, Hooke thought. He decided to take a look. Hooke trained his microscope on a cork, and what do you think he saw? Nothing. It turns out that microscopes do not work very well on reflected light. Perhaps he had better try again. So he took out his penknife and cut a very thin sliver of cork and shined a bright light through it. Then when he peered through the microscope, he had see something, and it puzzled him. Hooke wrote at the time that the cork seemed to be composed of little boxes. The little boxes were filled with air, and that's why corks float. He called the little boxes cells because they reminded him the rows of monk cells in a monastery, and a new scientific field called microscopy was born. The group that week was pleased with the Hooke's demonstration, but a full century would pass before the scientific world grasped the meaning and importance of Hooke's cells. One of the first people to move on the idea was the German naturalist Lorenzo Oken, who stated in 1805, all organic beings originate from and consist of vesicles or cells. But in spite of this rather clear and encompassing statement, the formulation of the cell theory is usually credited to two other Germans, the botanist Matthias Jacob Schleiden and the zoologist Theodor Schwann. In 1839, they published a conclusion that they had reached more or less simultaneously. They said that all living things were composed of cells. Either they were better at public relations than Oken, or perhaps the world was simply more ready to listen in 1839, but Schleiden and Schwann got the credit. Thus, Schleiden and Schwann are jointly credited with the having originated the cell theory. 50 years later, a fourth German, Rudolf Virchow, had boiled down the doctrine to all cells arise from pre-existing cells. To survive, each cell must be able to carry out all functions necessary for life. It must be able to absorb, transport, digest, synthesize, respire, excrete, reproduce, and respond. So much for such a little unit of life. To understand how this is possible, keep in mind that all cells are enclosed by cell membrane. The cell membrane is composed largely of proteins and lipids. The cell membrane separates the content from extracellular fluid. The cell membrane forms cell's boundary. The function of the cell membrane is to control the movement of substances into and out of the cell. The cell membrane provides a homeostatic or balanced environment inside the cell. The cell membrane is semi-permeable, or another way to say, selectively permeable. It lets certain things get in and out while stops other things from moving freely through itself. Please remember this characteristic. There is no quiz or exam that does not ask about this characteristic of cell membrane, if the test, of course, is on cell. Semi-permeable literally means relatively permeable. Semi means somewhat or partially, and permeable in Latin means to pass through or to go through. Phospholipids are key constituent of cell membranes. The molecule of phospholipid composed of glycerol, a phosphate group. You cannot see this on this diagram, but keep in mind that this phosphate is negatively charged, making the head polar and hydrophilic or water-loving. Another part of the phospholipid molecule is two fatty acid chains, or we might call them tails. One tail usually has one or more double bonds. That is, It is unsaturated. Remember from the lecture 9, double bonds form a kink in a molecule, and you see this here in this diagram. While the other tail does not have double bond and in its chain, and therefore it is straight tail. We call such lipids saturated. What are they saturated with? You're correct if you say with hydrogen. The tail saturated with hydrogen. Phospholipids are amphipathic molecules. In Greek, amphos means both, and pathic means feeling. Of course, molecules do not have feelings, but it implies that the phospholipid molecules has two contrasting characteristics. On one hand, it is hydrophilic, and on another hand, it is hydrophobic molecule, a sort of molecule with a double personality. The heads are polar region of the molecule. As you remember, polar molecules are hydrophilic molecules. So in water, their heads turned towards water molecules. By the way, why are they polar? Because they contain what? Phosphate group, negatively charged phosphate group. Their tails are non-polar, hydrophobic regions, water-hating regions. If exposed to water, they hide from the water molecules. If phospholipids from the cell membrane are purified and added to water, they will spontaneously form into membranes that look very much like cell membranes from which they were extracted. The hydrophobic fatty acid tails of the phospholipids will face towards each other. And the charged phosphate heads will face towards the water. Membrane is held together by hydrophobic interactions. These interactions are weaker than covalent bonds. Cholesterol restrains the movement of phospholipids. So the membrane is not too fluid, but as temperature decreases, it is the cholesterol that prevents membrane to solidify. We find cholesterol only in animal cell membranes, not plant cell membranes. But plants contain related compounds, sterols, that fulfill a similar function. Again, the sterols allow the membrane remain fluid as temperature decreases. Membrane must be fluid to work properly. If membrane solidifies, its permeability changes, and this leads enzymatic proteins to become inactive, and as a result, cell dies. On this slide, you see proteins that penetrate the hydrophobic core of a phospholipid bilayer. I believe you understand the etymology of the name. Bi means two or double. The membrane consists of two layers of phospholipids. A membrane is a collage of different proteins embedded in the fluid matrix of phospholipid bilayer. 50 kinds of proteins have been found in the plasma membrane of the red blood cell. Because of this mosaic arrangement of proteins and lipids and the fact that membranes are fluid, the model that you see on the slide called fluid-mosaic model of the plasma membrane. On this slide, you see proteins that penetrate the hydrophobic core of the phospholipid bilayer. We call them integral proteins or transmembrane proteins. Integral meaning pertaining to a whole. Extrinsic belong as a part of a whole. Transmembrane, trans means across or through, across membrane, through membrane. Transmembrane channels, also called membrane channels, are pores within the phospholipid bilayer. The channels are formed by integral or transmembrane proteins, and they cross the cell membrane connecting the cytosol or cytoplasm of the cell to the extracellular matrix. Another types of proteins are peripheral proteins. They are not embedded in the phospholipid bilayer, but loosely attached to it. Besides transport, the proteins that we just looked at have many other functions in the cell. One of them is enzymatic activity. Many of the proteins within the cell membrane are enzymes, a biological catalyst. Almost all metabolic processes within the cell need enzyme catalyst in order to occur in rates fast enough to sustain life. Changes in protein level, protein localization, protein activity, and protein-protein interactions are critical aspects of signal transduction, allowing cell to react highly specifically to nearly limitless set of cues and other to vary the sensitivity, duration, and the dynamic of the response. Intracellular junctions are specialized regions of contact between the plasma membrane and adjacent cells. They are essential to any multicellular organism, providing the structural means by which groups of cells may adhere and interact. Structural proteins help cell to maintain its shape. Cell recognition proteins or glycoproteins have several functions, such as recognition of pathogens. These proteins are also serve as an identity badge or a passport. The membrane has a phos component to addition of the phospholipids, proteins, and cholesterol. It is carbohydrates. You see them on my diagram like green little trees on the surface of the cell membranes. Some of them attach to the proteins, and the whole structure is called glycoproteins. Some of carbohydrates are attached to the phospholipids, and together they called glycolipids. The familia A and B blood group types are due to cell surface carbohydrates. On this diagram, I labeled for you glycoprotein and glycolipid. I also drew fibers of extracellular matrix. Collection of glycoproteins, glycolipids, and carbohydrates on outer surface of the plasma membrane is called glycocalyx. Function of the glycocalyx includes lubrication and producing the cell membrane and identification of the cell for the immune system. Also, glycocalyx has receptors for hormones. What we have recently learned is that cells able to recognize each other on the basis of cell structure. The clue substances are both carbohydrates and proteins. The cell recognition is important in many processes, including embryological development and immune responses. In one rather interesting demonstration, cells from different types of tissue were mixed together on the appropriate nutrient medium and have been left to see how they will interact. The results are surprising. Each type appears to move about and seek out others of its kind in such a way as to form individual masses of specific tissue type, presumably because each type of cell membrane has specific proteins and receptor molecules that react to these proteins. Cell surface protein called antigen also enable protective white blood cells to recognize and destroy most cancerous cells. And you undoubtedly have heard about the difficulties of rejection in organ and tissue transplants. Foreign tissues are rejected because their surface proteins and carbohydrates are not recognized by protective white blood cells, so they destroy just as any invader of the body would be. Not part of the cell membrane, but physically attached to its inner surface is cytoskeleton, a network of microtubules and microfilaments that give the animal cell its shape and its ability to change that shape. The cell membrane is like a butter smeared on the surface of the cytoskeleton framework. Please remember this important characteristic of the cell membrane I just described. That is, cell membrane is semi-permeable and the cell membranes are fluid. If cell membrane solidifies, cell dies quickly. As you can easily imagine, environment around the cell is constantly in changes. So what changes around the cells? Changes of temperature, of chemical composition of the environment, including, of course, amount of moisture, various amount of pressure. In order for cells to be alive, cells have to maintain homeostasis. Homeostasis is covered in lecture number 2. And since the cell's membrane regulates the movement of molecules in and out of the cell membranes play important role in keeping homeostasis of those cells. The laws that describe the movement of molecules help us to understand how homeostasis reached. Therefore, in our next portion of this lecture, I will describe two major processes: passive transport, that is diffusion, facilitated diffusion, and osmosis, and briefly I will introduce you to active transport. So please join me for part 2 of this lecture.